So it is my pleasure today to introduce today's speaker for our departmental grand rounds, Dr. Leticia Oliveira-Ferrer. She'll tell you how to pronounce her name properly because it's a Spanish name. She's speaking to us today from the University Medical Center in Hamburg, Germany. Her background includes studying chemistry at the University of Oviedo in Spain. She then received her PhD in organic chemistry at the University of Hamburg, Germany. And then she did what is called a habilitation, which in Germany, I'm understanding it's almost like a second doctorate. And this was in experimental oncology and biochemistry at the medical faculty of the University of Hamburg. Since 2016, she served as the head of gynecology research lab in the Department of Gynecology. And although I've never met Dr. Oliveira-Ferrer before, I was struck by a recent publication on the molecular characteristics and the tumorigenicity of ascites-derived tumor cells that was recently published in Molecular Oncology. And I personally found this manuscript especially interesting since my lab has done experiments with ovarian cancer ascites and spheroids, and her paper really answered many questions that have been in the ovarian cancer scientific field for years. So without further ado, the title of her presentation today is Mechanisms of Ovarian Cancer Progression, Tumor Heterogeneity, and the Role of Ascites-Derived Tumor Cells. So I'll let Leticia go ahead and share the screen and start the show. Thank you again for agreeing to be here, even though you're not with us today.
Yeah, thank you very much, Amy. Can you hear me, all of you, and see the first slide? I suppose, yes?
Yes, so that's great.
Yeah, perfect. Then I will start, and thank you very much for the kind presentation. And my name is Leticia Oliveira-Ferrer, so quite similar to how Amy pronounced it. I'm aware it's difficult. So I'm really glad to be here online and share with you a couple of research results from our laboratory in Hamburg, Germany. And as you see in the title, today I would like to talk a little bit about tumor heterogeneity, and especially about intraperitoneal tumor heterogeneity. And further on, go on a little bit deeper on the role of one specific compartment in ovarian cancer, in advanced ovarian cancer, which is the ascites and more precisely, the ascites-derived tumor cells. So let me start with introduction about ovarian cancer. I will keep it short, as I'm aware most of you are very familiar with this disease. Ovarian cancer is the most lethal gynecological malignancy with an expected 5-year mortality rate higher than 60%. And one reason of explaining this high mortality rate is that the disease is asymptomatic at early stages, and therefore around 70% of all new diagnosed patients show peritoneal metastasis at diagnosis. In contrast to other solid tumor entities, ovarian cancer disseminates primarily within the peritoneal cavity and not frequently hematogenously. Here you can see in the picture, frequently what we found are small tumor spots within the cavity, or metastatic spots in the peritoneal cavity. You see peritoneum, omentum, liver, or even at the diaphragm. The standard treatment for ovarian cancer consists of debulking surgery followed by platinum-based chemotherapy. Here, of course, the aim is to surgically remove as much tumor as possible. Again, the macroscopic tumor rest after surgery is still the most important prognostic marker for ovarian cancer patients. And of course, the subsequent chemotherapy aims to kill the remaining tumor cells. Unfortunately, recurrence and chemoresistance are observed in the majority of the patients. And here, many efforts have been done in order to identify a mechanism of chemoresistance, but also to establish reliable markers and signatures for therapy response. The majority of these studies have mainly considered the biology of the primary tumor from the ovaries or the tube. And although at the time point of treatment, the majority of the patients, as I already mentioned, has a tumor rest, which is not only arising from the ovaries or the tube, but also there are tumor rests from the well-established and maybe not completely resected metastatic sites. And in this context, our observations and those of others too have shown that the analysis of single samples in ovarian cancer provides a dramatically restricted view of the biology of this disease. Why is that? Ovarian cancer, and especially the most frequent histological subtype, the high-grade serous ovarian cancer, exhibit a prevalent genetic instability, mainly due to the loss of function of p53, but also of because of the deficiency in homologous recombination or other DNA repair pathways. So here, the combined dysregulation of apoptosis and DNA repair processes provides a mechanism, a very good mechanism for the generation of genomically diverse clones within the primary tumor, and leads eventually to this well-known intratumoral heterogeneity. So now we can imagine that these different clones within the primary tumor can independently detach from the tumor and be transported by the ascites or the peritoneal fluid and attach in any side of the peritoneal cavity, leading to a new metastasis with different genomic backgrounds. And this is exactly what this study showed a couple of years ago. Macpherson and colleagues have performed phylogenetic analysis. They have used whole genome sequencing and analyzed 68 different samples corresponding to 7 patients, which are displayed here in the picture. And what they have done is to determine the clonal mixture comprising each single sample. And they found that the majority, as of the first finding, was to see what we expected, that there was a very high clonal diversity within the peritoneal room. So the peritoneal metastasis, the peritoneal tumor tissues were compounded by different clones, but each of these samples were mainly clonally pure or contained clones from the same phylogenic clade. However, each patient contained or had at least one tumor sample that were polyclonal. And what they then conclude is that from these 7 patients showed here, 5 of them, 1, 2, 9, 4, and 7, showed a monoclonal and unidirectional seeding or dissemination, meaning from the primary tumor, one clone directly to one peritoneal site, whereas patient 3 and patient 10 mainly showed a polyclonal seeding and reseeding, so indicating that this tumor had a maybe more aggressive phenotype and more migratory phenotype. So what they found was also interesting, that was association between these different dissemination patterns and this different clonal distribution with the outcome of the patients. Although here we must be a little bit careful, the authors also remarked this, the patient cohort is too small to make this a statement now. So, but important is this clonal diversity within the peritoneal at the genomic level that was described by Macpherson and colleagues could be also observed at the transcriptome level in our lab, in the context of RNA sequencing project, including 244 ovarian cancer samples. Here, tumor tissue samples resected from different localization, including primary and metastatic sites, corresponding to 14 patients were available and were analyzed. And here you see the Pca plot so showing a little bit so the transcriptome profile of these samples. The 14 patients are listed here, labeled with different icons, and additionally, I have labeled each sample with a P or M, depending of the sample was resected from the primary tumor, meaning ovarian or tube, or from another metastatic site. So you see here what we observed is in the majority of the patient, or for the majority of the patient, we see a clear separation between the primary tumor and the metastatic tissue sample. As you see, exemplary for these three patients I have marked here. And if you pay attention, please, to patient 4, here you see especially that in this patient we have 2 different samples from the primary tumor, one from the left and one from the right ovary, and they are localized here in the Pca plot near to each other, but clearly separated from the metastatic tissue samples, which were resected from omentum and peritoneum. So we have a clear separation in, as I said, in the majority of the patients analyzed here. And this Pca plot analysis includes around 500 genes with the highest expression levels. But if we now go down with the number of genes analyzed and consider, for example, only around 50, which is the amount of genes analyzed or included in such molecular subtype profiles or signatures, like as the one described by Konietzki or by Totti et al., that classified the ovarian cancer, and especially the high-grade serous ovarian cancer, in 4 molecular subtypes: immune reactive, differentiated, proliferative, and mesenchymal. So if we take this signature consisting of 50 genes described in this publication, we could very nicely classify here in this table, exemplary for 6 patients, our patients based on the material of the primary tumor here. You have 2 patients from the representing the differentiated subgroup, 1 mesenchymal and 2 immune reactive and 1 proliferative. Now, if we analyze the material coming from other sites from the peritoneal cavity, so the metastatic sites, we see that in 3 of these, for 3 of these patients, 10, 11, and 3, we have different subgroups depending which tissue sample we are analyzing. And you see remarkably here, patient 3, we have 3 different tumor samples, and depending which sample we choose, we would classify this patient in a different subgroup, molecular subgroup, which might represent a problem, because this signature was already used for also for prognosis and prediction. So this really difference in the transcriptome of tumor tissues from the same patient within different compartments can be in part be explained by the clonal diversity at the genomic level I mentioned in the slide before. But we know from studies in the last year that the impact of the microenvironment play a key role in this different phenotype. That means there is an active crosstalk between tumor cells and stroma cells, meaning endothelial cells, mesothelial cells, adipocytes, or immune cells. And we know this active crosstalk endows the tumor cells with a specific molecular characteristics in each of the compartments within the peritoneal cavity. So I think we can conclude that this high intraperitoneal tumor heterogeneity in advanced ovarian cancer might represent a real challenge in order to establish reliable predictive markers or novel therapeutic targets. And therefore, we have decided a couple of years ago to focus our research in only one compartment, a compartment what we think is really plays a key role during disease progression, because it's responsible for the development of new metastases. And this is the ascites. Ascites, as you know, is a malignant fluid that grows during disease progression, is also associated with poor outcome, and is mainly produced or is coming because the high likeness of the blood vessels and the disruption of the lymphatic vessels. And the ascites fluid contains not only tumor cells, but also a large amount of cells from other origin, like immune cells, mesothelial cells, and so on, as well as signaling molecules such as cytokines and chemokines. And this whole environment is acts or has a pro-tumorigenic influence, and where the cells, the tumor cells, can survive and grow and develop. And as you see here in the picture, showing this a little schematic dissemination pattern of ovarian cancer, you see here two primary tumors. I hope you can see my mice. You have the primary tumor in at the ovary or the tube, and here the cells can detach as single cells, but also as cell aggregates, what we call spheroids. And of course, these cells are then transported throughout the peritoneal cavity by the peritoneal or the ascites fluid, and can attach to any surface of the cavity, to the mesothelial monolayer, or omentum, or other organs, as I mentioned before. And these tumor cells, the single tumor cells, but also these aggregates, must first adapt to this new microenvironment. They must survive this loss of attachment, but also evade the immune system. And especially the spheroids here exhibit a survival benefit. It's thought that maybe because of the tight aggregation that leads to a low drug diffusion within these structures, or maybe due to the low proliferative rate of the cells within the spheroids, these cells have a survival benefit and are regarded as metastatic units. So they are mainly responsible for the development and the formation of new metastatic lesions. So the spheroid formation might represent a key element of chemotherapy-sensitive recurrence. And therefore, the main goal of our project was, and still is, the disruption of these tumor cell aggregates. And in order to increase the tumor cell chemosensitivity of the cells included in these structures, and additionally to block the attachment of these spheroids to the surface of the peritoneal cavity, again, in order to reduce the amount of growing metastases. So to address these two questions, these two goals, we have to first get a little bit deeper in the molecular characteristics of these structures of these spheroids or these tumor cells within the ascites. And to do that, we have started with a first approach using the ascites from ovarian cancer cells after surgery. We have centrifuged all the cells, and after a first lyse step where the red blood cells were removed, we put the cells in cell culture and with normal cell culture flask, and observed after a few days that part of the subpopulation of these ascites-derived cells attached to the bottom of the flask. This is what we call, and it has also been described before in the literature, adherent cells, so ADs, and the other subpopulation part of these cells remained floating in the media. This is what we call non-adherent cells, or NDAs. And these cells are showing a very low, or rather no, proliferation in culture. You see here some exemplary pictures of these cells. You see the adherent cells. These are they are quite nicely growing. They show a very homogeneous population, show what we thought at the beginning, a very epithelial morphology. And in fact, we were hoping these are ovarian cancer cells that we can culture and grow and analyze without problems. And in fact, it has been described in several studies that these cells are ovarian cancer cells. But we will see after that this is unfortunately not the case. So the other population, the NADs, you see here is rather heterogeneous. It's a mixture of single cells, aggregates, all kinds of cell shapes. And as I said, these cells, they didn't proliferate in culture, and even after 10, 14 days, in some cases, the viability was dramatically reduced, and we couldn't analyze them anymore. So then we started characterizing these two populations, and first performed Western blot analysis. We analyze here, as you see, 6 pairs containing ADs and NADs, and we look for different markers, some epithelial or ovarian cancer characteristic markers, such as Ekaterine, ALCAM, or cytokeratin 7, and use another marker, vimentin, which is a mesenchymal factor. And we saw, to our surprise, that the ADs, in comparison to the NADs, express very low levels of Ekaterine, ALCAM, or cytokeratin 7, but express very high levels of the mesenchymal marker vimentin. So we thought, okay, the ADs are not really tumor cells, so the epithelial cells maybe are located or are contained in the NADs subpopulation. And this was confirmed by a second method with cell flow cytometry using 4 antibody panel, including EpCAM for the staining of tumor cells, CD24 for the staining of tumor stem cells, CD90 for mesenchymal cells, and CD45 for immune cells. We saw first in this upper side that we have in both subpopulation quite large amount of cells staining for CD45, meaning a large population of immune cells. And the difference between these two populations was that CD90 positivity, but was mainly found in the ADs, in line with the data from Western blot. In the NADs, we found a CD90 negative and CD45 negative population, which was quite pure positive for CD24 and EpCAM, indicating these are most probably ovarian cancer cells. So we can see that in contrast to our expectations, the tumor cells do not attach to the bottom of the flask. They remain floating as non-adherent cells. We enlarged a little bit the FACS panel and added two markers, podoplanine and mesothelin for mesenchymal markers, and mesothelin for trying to identify possible mesothelial cells, and also two markers, CD44 and CD133, which are markers for stem cells. And in fact, we analyzed several samples from different patients using this panel, and these slides show one representative example from one patient showing, but the same trend I showed you in the slide before. ADs here, again, two different pictures of these, doesn't contain or don't contain any tumor cells, EpCAM or CD24 positive cells. Rather, they are CD90 positive and podoplanine positive. No mesothelin expression was found here, and subpopulation was also positive for CD44. The NADs, again, very clear positivity for tumor cells, EpCAM and CD24, Interestingly, a subpopulation was positive for mesothelin and very few cells with the stem cell marker CD44. So we could conclude that the ADs were really highly proliferative in vitro, but they were not, or they didn't include tumor cells. So this could be in part confirmed by the in vivo analysis. We have the opportunity to inject 7 samples, 7 ADs culture for a few weeks, and in SCID mice, so immune deficient mice. And after around 7 to 9 months, we did not detect any tumor development, any intraperitoneal carcinosis. So we can conclude that these cells are not tumorigenic in vivo. For the NADs, the injection was not possible because, as I mentioned before, these cells are not really proliferative, and after a couple of days or a couple of weeks, they are not anymore viable. And so we can't get enough cells to get injected into the mice. So this first approach gave us important information, that's right, but didn't brought us forward in order to isolate the tumor cells with aim to characterize them. So what we did in a second approach was to analyze the cells at the very beginning. So of course, we first performed the first step, the lysis, to remove the red blood cells, but then we took the cell pellet after centrifugation and perform a filtration step with a cell strainer with pores of 30 nanometers, could nicely separate these two subpopulations, the one with a bigger aggregates and the small one containing only single cells. Here you see some pictures. The original cell population is a very heterogeneous mixture containing single cells and spheroids. The large fraction contains a very pure spheroid population, and the small fraction also very pure single cell population. And now, after characterization with cells flow cytometry, using the same panel I mentioned before, meaning CD45, immune cells, mesenchymal cells, CD90, and tumor cells, CD24 and EpCAM, we could observe that in the original cell population, we have a large amount of cells, immune cells, but in this example, also lot of tumor cells. I must say this is not always the case. We have analyzed until now around 80 samples from different patients, and I have to admit the heterogeneity between patients is also very high. Sometimes you have very much immune cells and less tumor cells, and other cases are like this one displayed in the slide. But what we can say is after this filtering step, we have always an enrichment of immune cells and these mesenchymal cells in the small fraction, and reduction in the amount of tumor cells. And we have in the large fraction an increase and enrichment in the tumor cell population. As you see here, we have around 90% tumor cells. And of course, we have a couple of CD45 positive and CD90 positives, but these results indicate that the majority of the tumor cells in the cell ascites-derived cells is in these spheroids. And this could be confirmed using other techniques like immunohistochemistry and immunofluorescence. You see here for 3 different patients, the tumor cells and the spheroids are EpCAM positive and also E-cadherin positive. And we have some signals for CD45 immune cells that seem to be not floating around, but rather embedded or associated with these spheroid structures. And the same results could be obtained in immune fluorescence experiments. You see here very nicely EpCAM staining for the tumor cells within the spheroids and single CD45 positive immune cells associated to these spheroid or aggregate structures. And the behavior in vivo was very good. As expected, these cells were highly tumorigenic. If you have a look here in this table, in this second position, you see from the spheroids, I mean the injection of the large fraction, we have 90% of the mice developed carcinosis and 70% developed ascites, including also tumor cells. And in comparison, when we injected the original ascites-derived cell pellets, meaning the heterogeneous mixture of single and aggregates, then we have much less or a reduced efficiency in the development of carcinosis. You see here, only 50% developed carcinosis and 20% ascites. What was also very interesting was the dissemination pattern observed in the mice. You see here most of the mice showed a mealy-like dissemination pattern, showing these small tumor deposits all over the peritoneal cavity, similar to what is observed in surgery. And two mice showed this non-miliary-like spread pattern. This was a rather bigger tumor mass bulks, mostly associated to the ovaries. And one mice developed only ascites, not solid tumor could be observed. The ascites was, but included high density of tumor cells. So what was really interesting here was that we found really a strong similarity in the tumor dissemination pattern observed in the mice and in the corresponding ovarian cancer patient as described in the surgery protocol. So this let's suggest that this model, this in vivo model, might represent a promising tool for analyzing ovarian cancer, the biology of ovarian cancer, but also therapy response in a more personalized manner. And the next step was, of course, to to characterize these spheroids. Sorry, and in order to do that, we perform RNA-seq analysis. We analyzed 10 ascites-derived spheroids and compare the transcriptome with the transcriptome of the solid tumor tissue. In this case, we choose to 3 different groups containing or including 10 tissue samples that were chosen based on patient age, FIGO stage, and histology matched to the ascites-derived spheroid group. And you see first in the Pca plot nicely separation between the ascites-derived spheroids here in pink, and the other samples, the tumor tissue samples, on the red and the right side. And we only considered, when we compared the regulation between these 4 groups, we only considered commonly deregulated genes in these 3 different comparisons and came up to 260 upregulated genes in ascites and in comparison to tumor tissues, and 600 downregulated genes. And among the upregulated genes, we found several genes related to chemoresistance, also a couple of them associated with cell adhesion, cell barrier, and tight junctions. Also 3 transcription factors, 2 of them were well known in our lab from previous studies, FOS and Jun. And also many genes associated to the oxidative phosphorylation pathway. We also find upregulation of 2 macrophage-related genes. On the other hand, genes downregulated in ascites in comparison to tumor tissue were mainly two pathways, angiogenesis related and extracellular structure organization associated. This is something that one might expect since the spheroids don't have any blood support, any blood vessels, and they also don't produce not much extracellular matrix in comparison with the established in the solid tumor samples. And in the next step, we focus for the first on the oxidative phosphorylation. Oxidative phosphorylation, as you know, is a metabolic process that takes place in the mitochondria of the cells, and in which the energy supplied by nutrients like glucose during oxidation is used to synthesize ATP molecules in a very efficient way. And this process takes place in differentiated tissue under the presence of oxygen. In the absence of oxygen, the cells rely on glycolysis for energy production. And this is a process which is much less efficient, as you see here, and leads to a high lactate production. In tumor tissue, the situation, the metabolism process is quite different. In proliferative tissues on tumors, we observe, as has been described, a metabolism process which is called the Warbler Effect or aerobic glycolysis. And in this process, regardless whether oxygen is present, cells seem to reduce the oxidative phosphorylation and shift to glycolysis for energy production with the accumulation of lactate. And this Warbler effect has been described for many solid tumor entities, including ovarian cancer. So we see that we have this aerobic glycolysis primarily as an energy production mechanism in the primary tumors, but also in the metastatic sites. And what we have seen in our Rna-Seq data is that the spheroids in the ascites behave different, induce a metabolic switch, and upregulates the oxidative phosphorylation process, as you see here. And we don't know exactly why. Maybe because these cells are very low proliferative and resemble the original differentiated tissue. Maybe that's the case. This is something we are currently studying. And what we have done in order to validate the Rna-Seq data is to look at the mitochondria. On one side, we looked at the mitochondrial mass, the amount of mitochondria, and the functionality with mitochondrial membrane potential marker. These are two Mitochondria green and red, and we could quantify it in FACS analysis for 9 ascites-derived spheroids, as you see here in this panel. And for 3 samples, we have matched pairs where we could compare the ascites-derived spheroids with the corresponding tumor tissue or from tumorides isolated from solid tumor. And you see here in the ascites spheroids, the majority of the samples show a high rate of cells with high mitochondrial membrane potential, indicating a high mitochondrial functionality, except in 2 samples from patient 10 and 11 that show lower values. If we compare now the ascites-derived spheroids with the corresponding tumorides, we see that the spheroids always have a higher rate of cells with high mitochondrial membrane potential in all 3 comparisons. In the immune fluorescence, we were also able to stain with these 2 markers, green as the mitochondrial mass, and red the mitochondrial membrane potential, so the functionality of the mitochondria. and we performed this analysis in ascites-derived spheroids as well as in tumorates. What we saw observed is that in the tumorates, this staining is homogeneous. We see functional OCFOs also in tumors, but the difference in the ascites-derived spheroids is that we have some sites which are extremely strong for these signals, as you see here or here, indicating that maybe this OCFOs upregulation is not really equally found in all the whole spheroid structure, but is maybe restricted to a certain subpopulation with the spheroids. This is something with now also addressing in our lab, and we are really looking forward the results in the next weeks. And in order to evaluate the effect of OCFOs inhibition on ascites-derived spheroids, we perform cell viability assays with 4 samples, with 4 ascites-derived samples from different patients after treatment with 2 OCFOs inhibitors, metformin and EACS 010759. Alone or together with cisplatin. Among the 4 patients showed here, 2 patients, 7 and 14, showed OCFOs inhibition response. As you see here, the second and the third bar showed a significant reduction of viability, cell viability after treatment with metformin and the second inhibitor. And the same for patient 14 here and here. In patient 7, we have additionally an increase in or a decrease of viability when we combined cisplatin and the OX4 inhibition in comparison with each treatment alone. 2 of the patients, 11 and 10, didn't show any response after OX4 inhibition. And although they were sensitive to cisplatin, as you see here. Interestingly, these 2 patients, 11 and 10, were the patients that showed very low rates of mitochondrial membrane potential in the FACS analysis, suggesting that these samples have very small population of cells with high mitochondrial activity, with high OX4 activity, and therefore, if we inhibit this pathway, the effect on viability will be not significant as we really observed. So, taking together all the information I have present today, we could say that the high intraperitoneal tumor heterogeneity in advanced ovarian cancer represents a real challenge in order to establish reliable predictive markers or novel therapeutic targets. Therefore, ascites, and especially ascites-derived spheroids, are very attractive, and they represent a very pure tumor cell population with a high tumor genetic tumor in vivo. Sorry. Ascites-derived spheroids show several specific molecular characteristics that might be suitable as therapeutic targets in advanced ovarian cancer. Of course, further analyses are needed in order to validate the findings from the Rna-Seq analysis and their functional relevance on ascites-derived spheroids. And these analyses are currently ongoing in our lab. We are not only working on OX4s further, but we are also analyzing other chemo-resistant associated genes and also some cell adhesion molecules which might be involved specifically in this step of attachment and adhesion to the peritoneal wall. Yes, and with this slide, I would like to finish my presentation and thank all the members from our research laboratory in Hamburg, especially Yi Ding, who performed all the experiments on ascites cells, and Karen Lekla, that were pretty much involved in the sequencing analysis. And of course, the head of our department, Professor Barbara Smalfeld, who is always taking care that we receive enough material in the lab from the clinic in order to perform this analysis, and our cooperation partners from the Institute of Anatomy, who helped us with the mice experiments, and of course the bioinformaticians that were involved in the analysis of the RNA-seq data. And thank you very much you all for your attention, and I would be very glad to answer your questions.
Great. Okay, well, thank you very much for the great talk. It is open for any questions people have. If you want to raise your hand, they can try to find you. I'd like to just start out with one question, which is, you had mentioned you've already done this separating the ascites cells, the large spheroids, from the single cells for 80 patients, and have done some analysis on them. And I was just curious if you have been able to do any clinical correlations with some of this data that you have, like the patients you mentioned that have lots of immune cells but very few spheroids, that sort of thing. Is there some correlation there you found with, long-term survival or anything like that?
Yeah, this is really interesting, and we are working on that. But the problem is we started this project 2.5 years ago. So the patients are still in the first phase of the disease. So we have not a long follow-up to correlate with this data. But we have some data on recurrence and quite preliminary results, but it's nothing I could really tell to be sure that it has to be validated, and we have to to analyze longer follow-up data in order to be sure. And concerning the immune cells, it will be also interesting to look a little bit deeper which immune populations are involved there, because CD45 is quite a broad marker, and so that's the reason we are now looking a little bit deeper, differentiated different T-cell subsets, and also macrophages are quite interesting. It's something it's going on now, but we do not have any correlation data until now.
We have a question from Tim Starr.
Hi, thanks. Very nice presentation. It was interesting how you started out saying that there are a lot of clones in ovarian cancer, and so you opted to go with ascites. And I know, I think only about half of ovarian cancer patients have ascites. And I'm wondering if you looked at your clones, if you looked at your ascites, and if you could find the same clonal diversity in your ascites that was found when you look at different metastatic sites.
Yeah, thank you for this question. This is, you are completely right. I didn't look at the clonal diversity in ascites. We didn't perform genomic analysis on that, only transcriptome analysis. But I'm quite sure we have a clonal diversity in ascites. That is for sure. At the genomic level, what I was hoping is that since the microenvironment is common for all the cells swimming around, is that at least the transcriptome is common for all of them, not completely common, but at least is more uniform, and is something we can target more specifically. But it's also not clear. What we are now starting is a project where we will do single cell sequencing. Single cell sequencing will give us much more information about that. If we have diversity within the ascites, because we have seen this RNA-seq data, it was a beginning, but it's always a problem. You see all average, and you don't know which cells exactly upregulate OXPHOS, for example, or other markers. So single cell, I think it will help us to identify subpopulations and to see how broad is this diversity you are asking there?
Yeah, we should talk offline sometime. We've done single cell now on about 60 patients, so it would be great to collaborate. Not on ascites though, only on
not on
tumor.
ascites. Okay.
Correct.
Oh,
correct.
that's really great, because we have no experience on this technique, and I know it's it's quite challenging. So I'm very happy.
All right.
Thank you.
Great. Are there other questions? Because I just have a semi-technical question, which is regarding the flow cytometry of spheroids. You had said you did flow cytometry of spheroids. It just seems like when I've tried to do it, the flow core facility won't allow me to plug up the machine. So I was wondering, do you dissociate them at all, or do you have a different machine that allows the change?
No special machine. It's a normal one, but we dissociate the spheroids with acutase. I don't know if this is a little bit softer than trypsin, and it depends. So it's really different from patient to patient. From some patients, the spheroids disaggregate quite nicely, and after a couple of minutes, some of them they are much more tight, and we have to incubate with this enzyme for, I don't know, 7 to 10 minutes, and then you get really a nice suspension and can analyze single cells. Yes.
Okay, so you're not concerned that the acutase removes some of the markers that you're actually looking for?
It could happen, yeah, of course. that's something it could happen. That's the reason we choose acutase instead of trypsin. At least for the cell adhesion molecules, it should be a little bit more softer or more.
Right.
Yeah. But you're right, that could happen.
Okay.
This is Andy Nelson. I actually work with Tim Starr. I'm a molecular pathologist, but I work with Tim on that single cell ovarian cancer project. And as you were describing your tumor spheroids and the increased oncogenic potential in the model, I was wondering, has your laboratory looked at any of the components of the ECM that helps keep that spheroid together? Like, I mean, is it rich in collagens, any particular subtypes of collagens, hyaluronan? Just thinking, is there something that creates essentially a portable microenvironment with those spheroids that may help even support some of the metabolomic, OXPHOS stuff that you've looked at?
Yeah, that's a really good question. We didn't look at that because what we have done until now is only to compare the spheroids with the tumor tissue. And as I mentioned, compared to the tumor tissue, you have much less extracellular matrix. This is quite sure. But you have right. We could be able to at least to stain maybe using immune cytochemistry or immune fluorescence, collagen, and so on, and look of hyaluronic acid. That's quite interesting point. Yeah, that's really a good idea to find something as useful to put the cells together. Yeah, that's a good point.
If you follow up with Tim, we can talk, and then Jim, who's my mentor and collaborator, just turned on his camera, so he's going to ask questions about that too.
Okay, great.
And I would like to just plug in that I did notice on your heat map that you did have some what you call barrier or cell, I don't know if you call them cell membrane or barrier proteins. So there were Claudin 4 is one of my favorites, that would cause these to have such tight adhesions to each other. Because we've tried like trypsin and acutase to try to get some of those patient ascites apart, and they're really tight. That's why I just like, oh, well, so I guess we need to go longer.
Okay, yeah. So we can exchange protocols. This would not be a problem. Good. Yeah.
And Jim, you got?
I do. I have a question. Thank you, and thank you for that presentation. It was terrific. So we're a cell adhesion laboratory. Have been involved in that for as long as Amy has been involved in cell adhesion. And you have these non-adherent, what I thought was fascinating, you have these spheroids that don't proliferate. You have adherent cells that do. Have you taken the spheroids and put them into the, along Andy's question, extracellular matrix that would cause them to adhere, change their shape, change their cytoskeletal organization, and so on, to make them proliferative, or to alter their tumorigenic phenotype? Or I mean, like, the question is there phenotypic heterogeneity based on how the cells are interacting with their microenvironment, just based on some age-old concepts like cell shape and cytoskeletal organization and pathway activation, that kind of thing? I mean, are they completely non-proliferative, or can you switch their phenotype by making them adhere?
Exactly. That's really, yeah, it's funny that you say that, because it's exactly what we are trying to do now.
Okay.
So what, okay, or at least if I understood your question right, correctly, what we are now starting to establish is a co-culture model in vitro. So we take these spheroids and put them, so if you put them in culture, they will not adhere very easily. But what we do now is we coat our flask with collagen,
Right.
and then mesothelial cells.
Exactly.
And culture them with ascites.
Exactly. And as Amy has shown, they can adhere to mesothelial cells and invade. And once if they are invasive and they do get in and change their phenotype, and just what you need is one more level of heterogeneity. But it would be interesting, I think, to see whether it is in fact, Andy's laughing at me, with me, not at me. Anyways, so terrific talk. Thank you so much.
Yeah, thank you.
I know, and we've tried to do something like what Jim's describing, only like 10%, if you took one patient's ascites spheroids and you let them adhere to mesothelial cells, like only 10% of them actually adhere and spread out and do invade, but it's a very small percent. It's like 1 to 5 or 10%. It's not like one patient has very proliferative or adhesive spheroids. It's very heterogeneous, as you described.
Yeah. This is that's really the big, huge difficulty in this disease. It's really so heterogeneous. From patient to patient, you have to establish a new protocol. It's really it's difficult. And what we have observed is in this first experiments we have done is this co-culture is. We have cultured these spheroids with different ascites from different patients. And there we observed different adhesion rates. So with some ascites, we had more cells adhered than with others. So there is something in the media, some factors there which are responsible to push this change, or this phenotypical change. And it will be interesting to find out what exactly this is. And it's maybe in one patient is more concentrated than in others. I don't know exactly. Yeah.
Oh, that's really great. So are there any more questions here? Okay, well, I would just like to, again, thank Leticia and let everyone know that if you do want to contact her, the email's not up there, but it's her last name, ferrer@uke.de. And she tells me feel free to contact her.
Of course, I'm really happy.
Okay, thank you very much. Really appreciate it.
Thank you very much, you all.